So in this section, what we're going to talk about are some cell cycle checkpoints, and you're just going to want to get out a blank sheet of paper of your own, and we're going to draw and fill in our own notes. Now, first thing that we need to understand is what is the idea of a checkpoint? A checkpoint is going to be some regulatory, either stages or steps in the cycle that must be passed or completed before the next step. Okay. So there are going to be a couple of checkpoints that we're going to talk about, but what we first need to do is actually draw our own cell cycle, and it doesn't need to be that big because most of the information we'll fill in is actually going to be on the outside of our paper. So here's my cell cycle. The cell cycle has several main points or main parts of it, and we're going to actually just divide it into, let's see if I can do this, 3 or 4 parts right now. So if you look at this, the first step we have is G1. Then we have the S phase. We have G2. And then we also have mitosis, which is M, and cytokinesis, which is C, and those can share a slot together. I'm going to use another color because I do want to indicate which part of this is going to have or is going to be a part of interphase. And interphase, as you should remember, consists of 3 parts, which is G1, S, and G2. And so all of this green right here represents interphase. All right, so what do we need to know first? The first checkpoint we're going to have is going to be between G1 and S phase. And between the G1 and S phase, it's literally going to be called, and I'll probably use a different color here, number 1. And I'm just going to write it over here. Number 1 is going to be our G1 S checkpoint. In this G1S checkpoint, what I'm going to have is basically I'm checking that growth has occurred properly and we can move into DNA replication. At this G1S checkpoint, we are also going to look at the environment internally for the cell and externally. And basically you're going to ask, is there enough material to do S? Is there enough material to replicate our DNA? So basically what that means is that we're going to actually need a signal here. And one of the signals that we're going to need is known as a CDK. CDKs will need to be present. So what is a CDK? Well, CDKs are cyclin dependent kinases. It's actually going to be a type of protein. The cyclin-dependent kinases, what they do is they phosphorylate, which means they change the shape, delta shape, change the shape of other molecules. And when they change that shape, they'll actually activate other molecules. And just something to keep in mind, the CDKs actually have many uses besides just acting as the signal or this go-between for the G1 and S phase. And they will increase in concentration during cell division. Okay? So next, what we're going to have is this G0 phase, G0. And so, if for some reason we don't pass a checkpoint, so I'll write number 2 over here. Number 2. If checkpoint is not passed, then a cell can do a couple of things. A, it could wait at G0. It can't divide, but can live or continue whatever its function is. B, the cell can amend or fix the problem and get necessary molecules to continue in cell division, so to continue going from G1 to S phase. Or C, the cell can go through apoptosis, which is known as cell death, especially if something besides just not having the right material is wrong with the cell. Okay? So it could go to G2, which is why I wrote, G0, G0, if for some reason it can't go on, but it can still function as the cell it is. From there, we have a couple of more checkpoints. We have what's known as the G2M checkpoint. And so we'll write this over here. And I really should just put a 3 right here. So we have the G2M checkpoint. At the G2M checkpoint, what we're going to do is check the cell, especially internally, for replication, accuracy.
Lily Banner, Sydney Cheek, Nia Baker, Matthew Barrera, Adrian Chasnoff, please see Miss Ellis in Student Services. Leslie Luia, Smith Pandya, Mark Nevsky, Jonathan Reyes-Diaz, Raven Cox, Keandra Miller, Roxanne Hills, Colby Allen, Tillman Chambers, Caitlin Chambers, Marcus Williams. Please go to Student Services. Alex Masson, Dwayne Gray, Ethan Freeman.
So at this third checkpoint, what we're going to do is, when we are in the G2 phase and about to move into mitosis, we're going to have the cell check itself, especially internally, for replication accuracy and has that occurred correctly. We're going to have a group or an enzyme or protein known as P53 that can check and correct any mistakes that occurred during replication. In addition to the P53 enzyme, we also have some proteins known as PMFs, which are maturation promoting factors. Factors. The MPFs are a group of CDKs. Like I mentioned before, CDKs have lots of different jobs, and this will actually encourage the cell to move into mitosis. So after our third checkpoint, we do have a fourth checkpoint that actually occurs in the middle of mitosis. And this is going to be our fourth checkpoint. In our fourth checkpoint, this is actually going to be known as the metaphase. checkpoint. And just like it sounds, this is actually going to check that spindle attachment to the kinetochore has occurred properly. Here we have another group of proteins known as the APC. The APC is known as the anaphase promoting complex. And just like it sounds, it's going to move our cell from metaphase into anaphase as long as the connection between the spindles and the kinetochore has happened properly. And as we know now, that's really important for the shortening of the spindle fibers so that we can move the chromosomes to the opposite ends of the cell. That's actually going to be our last checkpoint here, and from there we're going to be able to move into C, which is cytokinesis. Make sure you take some time to look over this and that you understand each of these steps. You should think about what is the name of the checkpoints or where the checkpoint's located. What are the requirements for us to actually pass into the next phase? Why is the location of these checkpoints important? And what happens if we don't pass the checkpoint? And that's all for today's notes.